Okay, so on the next slide, we see that we have a human chromosome, and part of that human chromosome is DNA. And now we have a picture of DNA, or a model of DNA, where you can see the base pairings. Every such little piece, cube in a way, it's not a cube, it's a cylinder, is what we call the base, right? Which we will talk about in further detail. And it has a matching one from a different color. So basically DNA is built out of 4 different kinds of bases that can be paired up. Okay? Through the process of transcription, an mRNA molecule or strand is produced, which is basically half or one side of the double helix DNA. So DNA has 2 strands that are complementary to one each other, complementary, and mRNA will be one of them. Okay, so for instance, in this case, you are shown in this slide the upper strand of the 2 strands of DNA. So I won't go into this too much because we will go into it in the future, but every mRNA molecule has an untranslated region that we will learn what it's all about. Then we have a start codon that we will also learn what it's about, but basically I'll just say that that's where translation starts. We have triplets of bases that are what we call codons, and each triplet is the codon for a certain amino acid, and we have a stop codon at the end. So the process of translation takes those codons on the mRNA that represent amino acids and actually combines them together to a polypeptide chain, which is going to be probably a protein at the end, but not only. Okay, we can see that there's 2 phases: in non-active polypeptide, which is generated first, and then an active polypeptide. Alright, so our DNA is full of untranslational regions, sometimes referred to as junk DNA. And we'll speak about junk DNA and see a few movies, so you understand that in this case junk DNA doesn't mean that it has no use. It's just a term and slang given to it before we learned a few more things. And you have a link to a movie that I would like you to see. So in general, we have here kind of a flow chart that shows that we have a gene or genes, right, that are the potential for a phenomenon. And then we go on to the level of execution, right? So if the execution actually does not come to be done, is not executed, then we might have an advantage or disadvantage. For instance, let's say that you have a gene that if it will be executed correctly, you will have a disease that will kill you. You still have the gene, but if it's not executed, meaning we're going to the right in the flow chart, that means you won't have the disease. So in this case, it is a mistake that the gene was not expressed, was not executed. But it gives that person an advantage because they're healthy and not diseased. But in case of a disease gene, a disease-bearing gene, if it is executed, it might be executed poorly, or perfectly, right? Once again, that might give you a advantage or disadvantage. The same goes for genes that include an advantage. Let's say something that would improve your physical abilities. If it's not executed, you might have a problem or lose the advantage intended. If it is executed, it can be executed poorly or perfectly, and then we don't know what the outcome would be. Alright, so basically those are the major ideas regarding genes. What happens with the execution and how does it relate to the information or the outcome that was intended initially. Okay, so we had 3 slides right now. One is a general idea. The second gives you a flow chart for a gene that is related to a disease, and the third one that I didn't go over because I think you already understand the idea is the idea of some kind of advantage. So we have the term of genetic mutations, which are called mistakes. They're called mistakes because they are believed to be random. I'm not so sure that they're actually random, and we'll talk about that in future chapters and presentations. So we have an addition or an insertion. That means that an added gene or sequence or base, meaning subunits of the whole gene, are added to the sequence. And once again, they can produce an advantage, a disadvantage, or perhaps have no meaning. It depends. We have a deletion, meaning that a part is taken out, either a whole gene or just a sequence or one base. We have what we call mutations that are called missense or nonsense. Okay, they're called also point mutations. So those are changes in the nucleotides themselves. And they can also be of sequence rearrangement. So we didn't change, we're taking a chunk or a sequence out of the gene and it's going to move to a different place. Okay? But it's changing its place within the gene, and it might have consequences. And what's called alien DNA or RNA means that something penetrates the body or the cell and stays there. Sometimes it leads to what we call symbiosis. So, for instance, the mitochondria is believed to be alien DNA or RNA that simply united with a human cell or with an organism cell, and through symbiosis started working together. So we have a slide describing addition or insertion. So you can see that the second sequence has one more amino acid added at the end, okay, relatively to the original. So the original is the upper strand or upper sequence, and the new one with the mistake, with the mutation of an addition and insertion, is the one on the lower part of the page. Then we have a slide about deletion mutations, right? So for instance, you can see that relatively to the first strand, 3 bases are deleted, meaning 1 codon, right? And the outcome is the last strand, the third strand on the bottom, that is 1 amino acid shorter than the example above. And then we have the missense, the point mutation. So basically, this is one change in a nucleotide. So from the length of the sequence of the strand is exactly the same, but one nucleotide within one codon is changed. All right, so you can see that one that is, let's see, 1, 2, 3, 4, 5, 6, 7, 8. The 8th nucleotide from the right side of the strand. In this case, it will be the 3', and we'll talk about that too, and we'll learn about that too. So it's just one nucleotide being changed. The nonsense point mutation, as you can see, is once again a change of one nucleotide, but what happens is that it makes the stop codon, that is the order to stop translating the mRNA into protein or to a polypeptide, is moved, so the product becomes shorter. Okay, and we have sequence rearrangement of one. We have one option here. So for instance, we have two codons on the upper strand that are just one after the other, and they're just changing places with each other. Okay, and then we have the second option where they're not close to each other and just changing one position rather than really moving farther away in the sequence. Okay, in this case, you can see that it affects the stop codon, and the product is shorter than what it should be, right? And then we have the slide for alien DNA. In this case, it's insertion number 1, and you can see that we have 3 genes and then an area of alien DNA that was inserted into there, and then the original proceeds with gene number 4 and gene number 5 and 6 and so on. Okay, and then we see a slide about the option of symbiosis. So this would be perfect to reflect or represent, for instance, the symbiosis between what was once an organism and has become an organelle in our cell, such as the mitochondria. That's a classic example. So I've just answered the next question, and the answer is the mitochondria. Okay, since I've addressed this next slide, I'm gonna skip it, and now we're talking about what does this all come down to. So one of the structures or components of a gene is called an allele. All right, and an allele is some kind of variation in the instructions or of the gene that will result most likely in a different trait. So we talked about abilities and skills, and we talked about something that has to do with appearance. Okay, so for instance, I have blue eyes. So I inherited the alleles, at least one for blue eyes, from one of my parents. I might have alleles for other colors of eyes, but this one is the one that was expressed. So in the gene that is responsible for the eye, the allele for color that was actually expressed, was actually executed, in my case, was for the color blue. Does that make sense? Alright, we'll stop here and continue from the next slide in the next video.